What kind of knowledge are we going to get today? Well, we're going to talk about the HP axis. Now recall that here we are in this last section of the course. We're talking about the endocrine system and the way that the endocrine system controls most of your chronic functions. We just got done with the control system of the nerves. Now we're talking about the control system of the endocrine system. And today we're looking at two topics: hypothalamic pituitary axis, and then an introduction to reproduction. As I mentioned in lab yesterday, you guys that are doing testis histology will be looking at the anatomy and physiology of male reproduction in vertebrates on Tuesday. We'll be doing female reproduction on Thursday, and then we'll go into putting sperm to eggs and talk about all the physiology surrounding pregnancy, and then that'll lead to lactation. So by the end of next week, you'll have all the information you need to assess your histology slides. Those of you that are doing thyroids and pancreas, you've already got the information you need, because we covered those earlier in the semester. So today, what I'm doing is covering chapter 76, as well as a little bit of additional information when I get to reproduction. We're going to go through the anatomical setup of the hypothalamic pituitary communication, which is actually twofold. Either the hypothalamus communicating with the anterior pituitary, or the hypothalamus communicating with the posterior pituitary. After that, we're going to move into one function of that HP axis control, reproduction. And I'll give you both a phylogenetic consideration of the way that sex determination occurs, and then a developmental perspective. We'll look both at the phylogeny and the ontogeny of reproduction. Okay, so this all begins at the level of the brain. Whatever endocrine pathway we're talking about, the majority of them begin at the hypothalamus. Now, we've already studied the hypothalamus a lot. We started early in the semester. We've mentioned hypothalamic control later on. Somebody give me something we've talked about already that the hypothalamus helps control. Okay. Osmolar homeostasis. Temperature, thermoregulation. Remember we have those thermosensors, right? That then downstream results in either vasoconstriction, vasodilation of the venous plexus. Talked about that. What's that? What do you mean the endocrine system? There are lots of releasing hormones that are released.
Isn't it like?
For instance, we talked about the thyroid.
Yeah, so I know I know somehow in the pathway it creates it helps create T3 and T4.
Correct. So you remember that when we talked about thyroid hormone system and metabolism? We said it all begins at the hypothalamus with TRH, that then goes to the anterior pituitary, TSH, that then goes to the thyroid. That's the theme I'm going to build on today, is this hypothalamic pituitary control of all the other endocrine systems. So let me start that with an image here for you, that a sketch of the lower part of your brain, so cerebral cortex, go deep down the cerebral cortex. We have a little, looks like a little P that extends from the base of the forebrain, still in the cerebrum. And up at the top would be the hypothalamus. And then extending down at the bottom, this little extension, little P-like structure dangling down, is the pituitary. Now I want to put pituitary in quotes here, because the pituitary gland itself is in structurally two different glands. There is a connective tissue separation. It's called the pars intermedia. That separates the anterior pituitary from the posterior pituitary. Don't get confused by whether I put the anterior or posterior on the right or left. It doesn't matter. It depends. You know, you'll see some textbooks the posterior being on one side. It is in reference to the face versus the back of the head. The name of the anterior pituitary is called the adenohypophysis. Let's look at the etymology of the phrase, the term adenohypophysis. Hypophysis is the name given to the pituitary. Whenever you read in a textbook the hypophysis, it is synonymous with the pituitary gland. The pituitary is the hypophysis. There's two parts of the hypophysis. One of them is glandular, adenohypophysis, whereas the other one is nervous, neurohypophysis. In other words, it is actually incorrect to talk about the pituitary gland. It's the same thing as talking about the adrenal gland. When I ask you, what does the adrenal gland do, Caitlin?
Multiple things.
You have to say, well, you tell me what part of the adrenal gland, right? Are you talking about the adrenal medulla or the adrenal cortex? They do very different things. The same thing is true of the pituitary gland. When I say, what does the pituitary gland do, you think, be more specific. Are you talking about the anterior adenohypophysis or the posterior? They are just as distinct as organs that would be in different parts of your body. And that's the way I want you to think about them. Let's start off then with talking about the adenohypophysis, the anterior pituitary. Developmentally, it begins in the pharynx. It is indeed epidermal ectoderm in origin. Those of you that take developmental biology next semester. How many of y'all going to take it? 2. All right, great. We're going to study this a lot. What we're going to see is in any vertebrate, a vertebrate begins as a solid, massive tissue that has 2 tubes on it. One of them becomes the spinal cord and brain. The other one becomes the GI tract. Here's what I'm telling you. In the early embryo, the tube that's the GI tract, up towards the mouth region and the pharynx region, has an outpocketing that migrates up towards the expanding cephalic brain. Remember those vesicles? We talked about expanding vesicles. As those are expanding, we have an outpocketing of the epidermal ectoderm. It is not part of the brain yet. Yeah, that's what I'm saying. It is glandular. It's not neural. All right, follow my logic here. There's those. Rathke's pouch is actually the name given to the evagination that's coming from the pharynx. The pharynx is glandular. It has glandular secretions. In other words, if you were to look at the histology of the anterior pituitary gland, it is going to look glandular. Let me just contrast it real quick with the neurohypophysis. The neurohypophysis begins as an outpocketing, as a downward extension of the hypothalamus. I want you to, in your mind right now, visualize the embryo. As we've got those primary vesicles forming in the brain, what also happens is the GI tube that will be your esophagus, your stomach, the GI tube has an extension that goes up. At the same time, your future hypothalamus has an extension that comes down. And they meet, and they fuse together, and they form this pea-looking like extension from your brain. One of them, epidermal ectoderm, the other one, neural ectoderm. It's two different organs that meet together and have a happy home for the rest of your life. And we call that home the hypophysis, the pituitary gland. So, let's now continue with our examination of I just wanted to contrast them before I went into the hormones and the cells and all that kind of stuff. They are different. I hope you see that by now. They're from different developmental origins. Let's go into the anterior pituitary. In general, when we contrast the functions of the anterior versus the posterior pituitary, the anterior pituitary has much slower responses when you compare them to the posterior pituitary. What kind of responses am I talking about? Well, these responses depend on exactly what kind of cells of the anterior pituitary are being interacted with by hormones that are being made from the hypothalamus. The way that the hypothalamus communicates with the anterior pituitary is by making a hormone that's put into the bloodstream that then travels to the cells of the glandular epithelium. Let me illustrate that. There would be a blood vessel that travels, a capillary, that travels from the hypothalamus down to the anterior pituitary. This blood vessel starts off as a capillary, gets a little thicker, it comes down to a capillary again down here. It's called the hypothalamic hypophyseal portal. The hypothalamic hypophyseal portal vein. Okay? Now, let's just take a time out. That's a lot of big words, but they explain exactly what it is. If you are a portal, any kind of portal vessel or portal vein, what does that mean? It's going from one place to another. It's transporting something from one place to another. Well, what's being transported here? Well, they're neurotransmitters that are going from the hypothalamus to the anterior pituitary. We're going from the hypothalamus to the anterior pituitary via the hypothalamic hypophyseal portal vessel. In other words, hormone number 1 is made up here in the hypothalamus. And it's made by these nuclei These are hypothalamic, hypophysiotropic nuclei. This is the name of the nuclei clusters that are up here. Again, don't get confused. When I say nuclei and I'm in the brain, what am I talking about? A bunch of cell bodies. Not near, a bunch of cell bodies that are clustered together. This is where we're going to make the first hormone. It's going to be released into the portal vein, and that hormone is going to travel down. All of these hormones are big proteins. They're big peptides. Talked about the classes of hormones last class. These are all big peptide hormones, and this is where they're going to bind to receptors on these cells of the anterior pituitary. Take a look at the handout. It says anterior pituitary. There's a table. Hormone number 1 are those hypothalamic hormones. Here are the options. Gonadotropin-releasing hormone. We abbreviate gonadotropin-releasing hormone, GnRH. Because it's a big old mouthful to say. So it's always referred to as GnRH, gonadotropin-releasing hormone. Gonadotropin-releasing hormone goes down through this hypothalamic, hypophyseal portal, and it binds to receptors that are on specific cells, and in this case, they're called gonadotropes. Gonadotrope is the name of the cell that's going to bind to the receptor. Notice that the anterior pituitary has several different cell types in it. We have gonadotropes, somatotropes, lactotropes, corticotropes, and thyrotropes. What's the difference in these cells? The receptors that they express. The first kind of hormone that's made I want to talk about are gonadotropin-releasing hormones. They go to the somatotropes, and in response, the somatotropes make, gonadotropes make hormone number 2. Now, hormone number 2 now, coming from the anterior pituitary, they're called gonadotropins. Because they're going to now go to the gonads, testes or ovaries. The 2 gonadotropins that we're going to be talking about exhaustively for reproduction are luteinizing hormone and follicle-stimulating hormone. Yes, the gonadotropes are the cells. The name, that's nothing but the name of the cell that releases, yes, gonadotropins, and the 2 gonadotropins are luteinizing hormone and follicle-stimulating hormone. Those are gonadotropins. Does that make sense, Rachel? Yeah, it's kind of like saying, gonadotropins is the generic term for LH and FSH together. Like saying androgens. Testosterone's an androgen. LH is a gonadotropin. Okay. Anterior pituitary. Let's talk about how in the world follicle-stimulating hormone is made. Somebody walk me through the process right now. I want you to describe the process through which follicle-stimulating hormone is made and released into the blood. Give me an explanation. Who wants to do it? Step right up, Brandon.
All right, so the hypothalamus, stemming from the hypothalamic hypophysiotropic nuclei, release a hormone, let's see, called gonadotropin-releasing hormone.
Yes.
And then,
where does it release it to?
It releases it to the anterior pituitary via that thing, or hypothalamic hypophyseal portal.
Yes, the portal vein.
Portal vein. And so in the anterior pituitary, there are cells known as gonadotropes, which are distinguished by the receptors that they express.
And they express receptors for
Gonadotropic
GnRH, which then, in response,
and they produce gonadotropins, their own hormone, and then so luteinizing hormone, follicle-stimulating
is
hormone.
released in the blood. Bam. It's a very complex system. It's slow. You tell me, why is it slow? We used to have multiple hormones that we made. We had to move it down through the bloodstream. Relatively slow process, okay? All right. I am not going to go through the others. You see them here. You all are intelligent individuals. You can read the names and the cells they come from. I mean, it makes sense. Thyrotropin-releasing hormone made by the hypothalamus, put into the hypothalamic hypophysiotropic, made by those nuclei, put into the hypothalamic hypophysioportal system, goes down to thyrotropes, which in response make TSH. Simple. All right. This is stuff that's being researched all the time. We're still learning about these cells. Who had this abstract? All right, Caitlin had this one. All right, Caitlin, let me just read the title, and then you give me a summary of what this abstract says. Embryonic development of gonadotrope cells and gonadotropic hormones. Lessons from fish.
Okay, so essentially, this study is looking at if we find out where like the gonadotropins are stemming from developmentally.
Developmentally.
And so what they had done, what they said that not much is known about it up until this point, and where the gonadotropins are stemming from. And so they.
Let's take a time out real quick.
Yeah.
We know kind of where they're coming from. You tell me, where would these gonadotropes, the cells making up the anterior pituitary, come from? The esophagus, they're coming from epidermal ectoderm. But we don't know the specifics. How in the world do they know exactly which cells they're coming from? Okay.
Okay, so the way they did this was they developed a stable transgenic line of Medaka.
Medaka, a fish.
Okay, and with an LH beta subunit gene, and so it's a promoter for luteinizing hormone.
Mm-hmm.
And they added a green fluorescence protein expression onto it so that you can see when it's expressing.
Okay.
And
GFP, y'all heard of that before?
yes.
I know the biochemistry students have. Exercise science students, GFP. Biology students, GFP. Okay. Biochemistry students, tell the rest of us what in the world we're talking about when we say GFP.
Protein that's essentially on a gene expression area, and when it fluoresces, that's when the gene is actually being expressed.
So, if the gene is being expressed, it looks like Cody's shirt. If it's not being expressed, it looks like Hannah's shirt. I mean, it's loser. Okay, let's stop right there. Here, let me just show you a little data that came from this recent study. It's a weird figure they made. These are the eyes, this would be the mouth of the embryonic Medaka. And what they're doing is they're showing that early on in development, 35 hours, 48 hours, 96 hours, what happens is we see these particular gene transcripts that are going to be characteristic of the gonadotropes later on that are being expressed. And indeed, okay, this is a little bit busy. Oh, man. Here's the results of the study. Don't get lost in the details. Let me show you. How do we get to a somatotrope, a thyrotrope, a corticotrope? How do we get to these distinguished gonadotrope cells? This will be a gonadotrope, right, in the anterior pituitary. These are going to be lactotropes. Well, if we begin with a template, that's the adeno hypothesis. Okay? We begin with epidermal ectoderm plate. And then we have differential gene expression of particular genes that are expressing proteins in a gradient from all FGF exclusively to only HH exclusively. And we have a gradient of those proteins cause the specific cellular differentiation into either a lactotrope, a corticotrope, a gonadotrope. So it's all about timing of expression of proteins in that plate, the adenohypophysis plate, that's going to lead to the future establishment of these gonadotropes or other cells. This happens very early in development. Here in this fish, by 72 hours, we've already got those adenohypophyses. Okay, that's the anterior pituitary. Let's turn the page and talk about the posterior pituitary, the neurohypophysis. As I mentioned earlier, it's neural in form. It functions very differently because look at the figure here. Instead of us having cells that are releasing hormones into a portal system, now we have a nucleus, either the paraventricular or the supraoptic, that then have an axon extending from them and terminating. The terminal end of the axon is the posterior pituitary. Make sure you get this. A posterior pituitary is nothing more than the terminal end of the axons from the cell bodies that began in the hypothalamus. It's an extension of the hypothalamus. I think neurohypophysis is a bad name for it. It'd be like, a better name would be hypothalamic annex. The hypothalamic annex. It's an extension off of the hypothalamus that goes down. Now, hormones that are released from this hypothalamic annex, that's got traction. Another band name. I got some more band names coming up today. Responses are very fast because it's neural in nature. It's not glandular, doesn't rely on multiple hormones. The responses are very, very fast in response. When we get to lactation, we'll talk about this specifically. Oxytocin is one hormone that's made by the posterior pituitary. When a woman hears her infant baby crying, she can eject milk like that. Does it take a long time? It's a neural response. The two hormones that are made by the posterior pituitary are oxytocin, which is coming from the paraventricular nucleus, and ADH/vasopressin, which is coming from the supraoptic nucleus. The paraventricular nucleus, let me take the time out, Rachel, make sure you know what I'm talking about. It's a lot of big words today, but when I'm saying a nucleus, what do I mean? A cluster of cell bodies. Notice how this is a bunch of cell bodies together in the brain. It's called the paraventricular nucleus because of the location of it. It's beside the ventricle. Duh. Pretty good name. Paraventricular nucleus, a bunch of cell bodies beside the ventricle in the hypothalamus. It has an axon, a group of axons that come down, and they release, in this case, oxytocin.
So are these hormones actually made in the posterior pituitary then, instead of?
Great question. Okay, these hormones are all polypeptides. You tell me, where are they made? They have to be made. Where in the cell? If it's a big protein, where are proteins made? In the nucleus of the cell. In other words, they are made in the nucleus of the cell. They are transported via axonal streaming down the axon. They are stored right here in vesicles at the terminal end and released as they're needed. It kind of sounds like neurotransmission, doesn't it? It kind of sounds like the way that large molecule neurotransmitters are made. And it is. It's the same thing. It's just now we're talking about it from an endocrine standpoint. Maybe you don't like it.
You said they're stored where? When they get to the terminal
At
end?
the vesicles, neurotransmitter vesicles,
They're stored the same
they're
way?
stored the same
Okay.
way. It's identical to when we talked about nervous communication.
I don't know. Yeah.
Sure?
Yeah.
All right. They're not small molecule neurotransmitters. They can't be made at the end. They have to be transcribed and translated up in the nucleus, moved down by an axonal streaming, and released at the terminal end. What's that?
It's like one big neuron.
It is! It is a big neuron. That's what I'm forgetting. Yeah, it's a nerve. It's nothing but a big neuron. A bunch of nerves that are going down and releasing upon stimuli a neurotransmitter. Now we're calling it a hormone node. The supraoptic nucleus is transcribing and translating ADH/vasopressin. Somebody explain to me why I'm saying slash. They're the same thing. It's the same molecule. It depends on what function we're talking about. Either a cardiovascular function, we call it vasopressin. If it's an osmoregulatory function, we call it ADH.
What is number?
With a limited number of tools in it. And we use those tools for different functions depending on, yeah.
Do both of those only express ADH or oxytocin, or those only two examples?
That's it.
Those only
Those are the only two. That's it. Posterior pituitary is pretty easy. Anterior pituitary, a lot more hormones, a lot more players. Yep. Okay. Now, I am going to be mentioning, as we go through the rest of the semester, the hypothalamic pituitary control of testes, the hypothalamic pituitary control of ovaries, the hypothalamic pituitary control of growth. And I expect you to understand what I'm talking about now with that adenohypophysis or neurohypophysis. Let's turn the page then, and let me give you a set for reproduction. We're going to talk about two contrasting modes of the way that sex determination happens in vertebrates. You are most familiar already with genetic sex determination. I'm also going to talk today about a topic I'm really passionate about, environmental sex determination. Let's begin with GSD. Everybody's familiar with this karyotype view. If you were to go in and do a karyotype of your chromosomes, here's what you'd see: 22 autosomes and one sex chromosome set. If you've got this little bitty Y with not too many genes on it, and a large X with a lot more genes on it, the outcome is going to be a male. If we have two X's, no Y, we're going to have. So what is this based on? It's based on the genes' hardwired expression of maleness or femaleness. You're familiar with that, and I'm going to give you the names of the genes in a few minutes. What happens early on in development, in other words, in animals that have GSD, is particular genes are expressed that dictate testis or ovary formation. We're going to be talking both about primary and secondary sex determination. Primary sex determination is whether or not we get a testis or an ovary. Secondary are all of the results of getting that testis or an ovary. So what I'm talking about right now is, how do we get this primary sex determination? How do we get the testis or an ovary? Well, in an animal with GSD, it's very easy. If you get the genes passed on from, in the case of mammals, dad, in the case of birds, mom, because I'm giving you way too much information right now. Here's some bonus. Everybody knows that mom is contributing the X chromosome, right, on the egg, and dad's producing either an X or a Y. In birds, it's opposite. It's not the male that has the two different sex chromosomes. It's the female in birds. So, just different means of getting two different sexes hardwired, birds or mammals. Let me talk though about animals that don't have genetic sex determination. Animals that let the environment determine their sex for them. And by environment, what I mean is either temperature or social cues. Probably my favorite stories about this have to do with some fishes that are inhabitants of coral reefs. These are wrasses, W-R-A-S-S-E-S. These wrasses are said to be protogynous. I don't expect you to have heard this word, but I expect you to figure out what it is. All right? You guys are getting really good at being able to go in and dissect a word for me. So, prefix proto.
Early.
Early or first. The females in the room should know what gynecologists do and what's gyn refer to? Female. Wrasses are all begin their life as females. And then all of a sudden, based on the social dynamics, some change into males. How in the world does this happen? We're still figuring it out. Who's got this abstract? All right, Claire, what does this tell us?
I spelled the brass wrong.
What?
I spelled brass wrong when I wrote it down.
That's the worst thing ever.
I know.
All right, Claire, what does this say?
They had two groups of female fish, and in one group they implanted estradiol, and in the other they implanted cholesterol. And the estradiol group couldn't undergo sex change, and they had high levels of aromatase mRNA.
Let's take a time out and just unpackage a couple of things that Claire said there. Why would they put cholesterol in based on last lecture? Because cholesterol is the precursor for all steroid hormones, right? They're putting estradiol in. Is estradiol at the end or beginning of the steroidogenic pathway? The very tail end. So they're kind of got both ends of the steroidogenic pathway. They're implanting cholesterol in some, and they're implanting estradiol 17 beta in others. And the ones that got the estradiol 17 beta, they couldn't become males. They're basically overriding some natural inherent conversion pathway. Could this have something to do with the aromatase enzyme? Okay. Talked about aromatase a little bit. Did I talk about last class? A little bit? The one difference between maleness and females, testosterone and estradiol, is expression of this aromatase activity. And indeed, we can alter this outcome. Okay, so here's what I'm telling you. We still cannot connect all the dots with how in the world the social cues, i.e., how many males are in the population, can determine whether one fish is going to become a male or not. But they do it, and they have to do it through the steroidogenic pathway. They have to do it by somehow turning off aromatase, perhaps, allowing more androgens to be there, allowing conversion from an ovary to a testis. Still don't know the details, though. We do know a lot more about the details of the way that temperature determines sex determination. So let me take a minute and talk about temperature-dependent sex determination. Most of you are probably familiar with this. But if I go out and I get 100 turtle eggs, and I bring them into the lab, I'm going to bring them into the lab, and down in 117, Animal Physiology Lab, we have 2 incubators. If I take 50 of those eggs, and I put them at 33 degrees Celsius, and I take the other 50, and I put them at 30 degrees Celsius, the ones at 33 will all be females. The ones at 30 will all be males. I have just engineered their sex. I have just 3 degrees Celsius. Crazy. In other words, what I just told you is temperature is determining the sex determination. Yep.
What is the evolutionary advantage of that?
Let's talk about that. All right.
Speed.
Here's a really interesting observation. I didn't give you this. It depends on the species as to what pattern they have. For instance, in Trachomys scripta, 27 degrees Celsius produces all males, 31 degrees all females. In sea turtles, like I said, 33 all female, 30 all male. Alligators, well, it depends on where we are as to how many males and how many females we get. Here's the adaptive value of letting the environment, as long as the environment's not changing really quickly. If the environment is changing over multiple generations, and you have temperature fluctuation in the environment, then it's called the pivotal temperature, the temperature that produces half males and half females, can float over time, still producing a Fisher's balanced sex ratio without having to hardwire it. In other words, it actually, over geologic time, produces better sex ratios than GSD does.
Can't you have multiple sexes in one nest
Yes.
dependent on how they're buried?
Yeah, but if you got 100 sea turtle eggs, most sea turtles lay 100 eggs. Those in the middle are getting the metabolic heat of all those around it. They're going to all be females. On the outside, you got males. So you got the males coming out first, then you get the females coming out. Crazy. One's deeper. This gets into all kind of cool ecological implications. Can a female sea turtle determine by the depth of her nest whether she's going to have males or females? This is very controversial. It's almost getting into you would want more females. Because investment-wise, that makes a lot more sense than the expendable males. So can we, by digging a shallower nest, produce more females? And is that happening in populations? Okay. What's the mechanism of this? Well, you give me a. What could be behind this? How in the world could temperature be determining maleness or femaleness? We got to have some kind of enzymes that are temperature sensitive. And indeed, did I give you this? I didn't give you this. All right. I'm not expecting you to memorize any of these genes. I just want you to know the concept. If you look at in mammals that are hardwired for sex determination, an ovary, this is the pattern of genes. SOX9 is reduced. SOX9 is increased in the testis. The reason for that is the Y chromosome has SOX9 on it. It's expressed. In contrast, the X chromosome has a lot of DAX1. The Y chromosome doesn't have any DAX1. DAX1 is reduced. Here's the way temperature works on an animal with TSD, is the same enzymes are increased or decreased, but now they're temperature sensitive. This works really well in ectotherms. This has led to a lot of questions. In other words, one good, easy softball question is, which came first, TSD or GSD? Did animals with GSD lose that ability, and then rely on temperature, or the other way around. Thoughts?
Just seems,
Ideas?
I would just like my train of thought would say that because the reptilians are like the ones we're looking at are more ESD, that I would say ESD probably first. Just
It's interesting when you look around today, and this has led to a lot of. When you look around today, many fishes have genetic sex termination. Many amphibians have genetic sex termination. Most reptiles have temperature. Birds and mammals have genetic.
yeah.
So you're like, well, fishes, well, the reason for that is because we're looking today, and we can't go back to when it originated, but we actually can. I love your reptile idea. That's where we're going. I got some snakes up here. Two different kinds of snakes. These are both taken. Both these pictures were taken within 15 minutes of the campus. One of them you do not want to approach. Which one?
Just stay away from both of them.
If you see a snake, walk away.
Let me. Hold on. Okay, here's a close up. This is the one you do not want to approach.
Your rule of thirds is off.
The characteristic of this viper, copperhead, around here is they have these pits and they have a slot right there on their eye. Here I'm going to tell you, if you get this close, it's too late. Do not rely on the pits or the slot to determine you're looking at a
I need to see its face. I have to get right next to it.
The hourglass-shaped, copper-colored. Stay away from it. All right.
What?
The top one is this cute little guy. Very friendly. Look at that. I mean, his eye is not intimidating at all. All right. This is a water snake. We got them in the college woods. We got them all over the place. It's a northern water snake. Here's where I'm going with this. Vipers, copperheads, rattlesnakes, are very recent evolutionarily. They are the most evolutionarily advanced group of snakes. These other snakes, colubrids, they've been around a long time. And there's even older ones, more ancestral. The most ancestral snakes are like the boas and the pythons. There's some on campus. Boas and pythons. Let's do a karyotype of the ancestral boas, the modern colubrids, the really, really recent vipers. And what we see is the vipers have distinct sex chromosomes, hardwired sex termination. When you go back to boas, pythons, they don't have any sex chromosomes yet. Now, they have genes that are there, but the sex chromosomes have not evolved enough to be distinct from each other. These guys, we know these boas are so ancient because they have pectoral and pelvic girdles. They actually have places limbs used to attach to. So these ancient snakes, here's the take-home message. These ancient snakes have just recently evolved genetic sex termination. It used to be temperature-dependent sex termination. And that's what the literature is showing us now. I don't think I've got an abstract on that. Okay. But bottom line, ESD was first, and now we have GSD. I want to take just a few minutes and talk about some implications of this temperature-dependent sex termination and some of the research that's going on in this area. And the first is a project that I was involved with, and it had to do with, by the way, we have just recently figured this stuff out. I mean, this is what I worked on just a few years ago. Not that many years ago. All right, I'm getting old. All right. Within your lifetime, we have figured this out, okay? And we're still figuring this out. I just told you that sea turtles, their sex, whether you get male or female turtles, is based on what? The temperature. Okay. Along the, let's see, I think I got a picture. There's Jared. Everybody knows Jared. This is my son, Jared. this summer, he's Cody's friend, right?
There's boats now.
Jared went to the beach with us on our beach trip. What I want you to notice is this is actually at Edisto. This is Edisto. Notice the color of the sand. Nice, dark colored sand. What happened on the Atlantic coast about 25, 30, 40 years ago is as the erosion was washing the sand away, we wanted to re-nourish the beaches. Pump in sand on the beaches to keep them from eroding away. Makes sense. You've got a beach house, you don't want to fall in the ocean. Where can we get sand from? Well, the first place they went for sand is right off the shore. And they would take these big barges, and they would take a big vacuum cleaner and pump the sand up on the beach. Worked good until we ran out of sand. Now, I kid you not, we ran out of sand. There wasn't that much sand out there on the shelf to do this. It was eroding away. Where can we go get sand? And on the Atlantic coast, specifically in Florida, on up into North Carolina, they've always been envious of the Gulf Coast sand. It's so white. Everybody likes that white sand. Why don't we just buy some white sand from the Bahamas? This is actually what happened. They pumped in white sand, brought it up on the beaches. This is what the sand should have looked like. This is again Edisto, a nice little beach crawl going up. And this is what they renourished it with. Beautiful white sand. For about 15 or 20 years on these beaches, sea turtles were coming up, laying their eggs, and 100% males were being hatched out. There wasn't a single female sea turtle that was hatched out. Why?
The white reflects.
The white does not absorb the light, it reflects the light, it's a lot colder. This is the same time, we were actually doing this the same time we were figuring out, wait a minute, turtles are temperature sex dependent. Then we started seeing that all those that are hatching out were males. We stopped using, thank goodness, sand that's not native to that area. Conservation efforts were tremendously impacted by our understanding of TSD. One of the ideas that's been batted around for a long time is, could dinosaur extinction be attributed to temperature-dependent sex determination? All right, y'all just had an exam about this apparently?
Stop.
It's a small one. It's very small.
Here's the idea. Big meteor hits. Big cloud of dust forms. Cool Earth. What if we had reptiles that were TSD and it cooled down and you were producing 100% males? Not good for a population, right? That's the idea. It could be one reason. Oh, I thought I had. Okay, good. Did somebody have this abstract? Dinosaurs? Ariel, what does this say?
This was basically looking at, was temperature-dependent sex determination a possible reason for dinosaur extinction? And they were thinking that maybe the climate heated up a little bit and caused 100% males to come out, and that's what caused the extinction.
Okay, so there's the idea.
So,
Climate change, all males, maybe those that had TSD went extinct.
so they went to the Hell Creek Formation in Montana, and they looked at 62 different species. 46 had genotypic sex determination, 16 TSD. They were completely wrong. The 16 that had temperature dependent, of the 16, 14 survived.
Yeah, the survivors had TSD.
Yeah. And in contrast, 61% of species with GSD went extinct.
Exactly. So, this idea of climate change causing massive problems on animals with TSD is not supported anymore. It was an idea. It was a sexy idea for a while, but it hasn't been supported by the data. Looking at the species that stayed alive, that didn't go extinct, they indeed had TSD. How could that be? Well,
Is it what you just said, where it's like changing over a long period of
Exactly.
time and they can adapt?
it's both that and
There-
there's a normal distribution for the pivotal temperature among a population. Some individuals will not respond exactly at 33 or 30. Some will be variants on that. And if that variation survives, they pass that variation on within a generation. Does that explanation make sense? Yeah. So it's like you have a normal distribution of any trait. This trait I'm talking about would be the temperature that caused you to go from maleness to femaleness. And if the one that survives is over here, it passes it on. So it's a flexible. It is what I was talking about earlier. It's very flexible for temperature change. This is currently the worry with accelerated climate change. Obviously, we're in a time right now of really rapid anthropogenic-induced climate change. Is that going to preferentially cause problems for animals with TSD? I think the population will be able. I'm not as concerned because of the history of the very rapid temperature changes that we've had and the fact that animals with TSD have survived. However, I am very concerned about the release of pollutants that are called endocrine disrupting contaminants. In the last 100 years, very recent, if you think geologically, I was giving a lecture yesterday to my non-majors, and I tried to get them to understand geologic timescale, which is impossible. I mean, 4.5 billion years. I'm like, you can't grasp that. It's kind of like grasping the national debt. I can't fathom it. I can, however, fathom a $10,000 debt, and I can fathom, instead of geologic timescale, I can fathom 365 days. I can fathom a year. And what I did with my students yesterday, my non-majors, is I said, imagine the Earth was created on January 1st. And then I said, it wasn't until mid-June that we had vertebrates. It wasn't until, here's where it gets really good. It wasn't until after Thanksgiving that we had mammals that we could really, and it wasn't until, here's the cool part. It's all above this, it's all here. It wasn't until New Year's Eve at 11 p.m. that we had Homo sapiens on the planet. In a calendar year. That's amazing if you think about it. How recent we are. What a flash of the pan. Here's what I'm telling you now. Over the last 100 years, New Year's Eve, 11:59, and right when the balls hit the ground, that we've released chemicals into our environment that can totally circumvent all physiological systems. The chemicals I'm most concerned about are those that are acting like estrogens, acting like androgens, decreasing receptors for your endocrine system, because it can circumvent the normal circuitry that we have. Give you a little background into this. I've already mentioned an alligator egg. If we take this alligator egg and in the sensitive window when gonads are formed, it's not the whole development now. It's not from day 1 till 60 days in incubation. It's during a sensitive window, whenever primary sex termination is occurring. If we take this egg and we incubate it at 33 degrees Celsius, we're going to get a male. If we incubate it at 30 degrees Celsius, we're going to get a female every time. If we put a drop of estrogen, estradiol 17 beta, and incubate it at this male temperature, we can make it be a female. Literally. We take a drop of estrogen, put it on there, Put it at a male temperature, it becomes a female. We have just overridden temperature's effect. There are lots of other compounds that can do that too. You probably don't recognize para-DDD. This is the breakdown product of DDT. We don't measure DDT anymore in our environment. We measure the breakdown product, which is an environmental estrogen. It's broken down. It's got a 100-year half-life, but it has DDD instead of DDT, what it was added as. Transnormichlor, another plasticizer. Lots of compounds can do this. Yep.
So in this kind of study, like all you have to do is expose them environmentally to estradiol, like the outside of the egg. What you're saying?
You have to give it in a diluent that's going to take a vehicle that's going to take it inside. So the estradiol has to get into the chorion membrane, but the way you do that is you paint it, and you put it in. We would put it like in DMSO. DMSO takes it right in.
I'm just thinking environmentally, like how it works, like natural.
Oh, good. How it works naturally is, yeah, organochlorines like DDT are lipophilic. Let's say you want to talk viviparous or oviparous. Which one do you want? Viviparous or oviparous? Live-bearing mammals. Anytime a female, a woman, lactates, she's mobilizing her fats. She's putting all lipophilic contaminants in her milk and delivering it to the baby. Milk is an excretion pathway in females. In animals that have eggs, oviparous animals, when they make the yolk is lipid. The yolk is protein and lipid. They mobilize their fat stores, i.e. DDT. They excrete it in the yolk, and it delivers it to the...
So it's the exposure to the mother.
Yes, it'd be maternal exposure, not the baby's exposure.
Okay.
Mom has bioaccumulated it through her lifetime. Really cool that so many cool things. Way too much to do. Females can excrete lipophilic hormones. Males can't. In any population, if you were to right now go and measure in an adult population contaminant burdens of males and females, the females have gotten rid of the contaminants the males have. And again, a lot of this endocrine disrupting research centers around this one enzyme. I mentioned this last class. I wanted to show you a picture of it. The difference in maleness and femaleness is that little fellow right there. We have aromatized testosterone with a hydrogen. And this causes every secondary sexual characteristic that we know of. So how can some endocrine disrupting contaminants work? Who had this one? Anna, what's this say?
There's two different studies. There's a descriptive study where juvenile alligators were exposed to contaminated leak and a control leak, and the juvenile alligators ended up showing significantly lower testosterone in their plasma.
Lower testosterone. Why is it lower? Where's testosterone going? Probably being converted to estrus, androgen estrogen through aromatase. So
there's a second study where hatchlings were exposed to two known endocrine disruptors and two herbicides. And so the endocrine disruptors affected the sex from male to female, but what they found for the herbicides is that it produced gonadal adrenal resinoprobe aromatase activity.
Yep.
That was characteristic of neither males nor females, so it's like a weird hybrid.
it's like, yeah. And you can, this is way too much. Don't worry about that. All right. Yeah, so instead of having normal female aromatase activity or normal male aromatase activity, let's stop right there. Which one's going to be higher? Aromatase, males or females? Females, right? Androgens, estrogens. Instead of it being really high or really low, animals that are exposed to this contaminant have this much. Not male-like, not female-like, expressing some odd amounts of estradiol, 17 beta. It's going to lead to altered secondary sexual characteristics. Currently, we're looking at other compounds like bisphenol A and phthalates. Both of them plasticizers. BPA, we've talked about before. Bisphenol A is this hardening agent in polycarbonate plastics. I'm going to be mentioning BPA a lot when we get to female reproduction and male reproduction. I just want to introduce you to now. It is being looked at in terms of one of the smoking guns right now, if you will, for a compound that's still a high volume compound released into the environment that has an effect, not lethally, but sublethally, altering the endocrine system of animals. We can see BPA, is it in the environment? Who had this one? Marley, what's this say?
Well, they took samples in just a normal, like, stream environment and in a, like to see one that was close to a landfill. And the landfill one actually had, like, slightly less BPA concentration in the water. They determined that it affects, like, it feminizes an embryo during neonatal like differentiation.
Yep.
It has to be more than there is in the environment
Yep.
naturally occurring.
Yeah. So, in the environment, can you find BPA? Can we go out and find BPA? Answer is yes. Where can we find it in the environment? Low levels. Low levels can be found in waterways all around. High levels can be found in landfill leachate, the water coming out of landfills. Somebody explain that to me.
People throw away plastic water bottles.
Plastic's not being recycled, end up in a landfill, it heats up, and it's actually the heat that mobilizes it from the plastic. It then rains, it washes it in the landfill leachate. Makes sense. Is this amount altering wildlife right now? No. Thank goodness. But it's at a critical threshold of doing that.
There are a bunch of major cities that are starting to require recycling. Like, you get fined if your your garbage man discovers that you're not recycling.
Why would you not? It's like, I mean, literally, it's like taking leaded paint and dumping it in the sewer. It makes no sense to throw away plastic in a landfill. It is introducing a contaminant into the waterway.
And in Canada, like, when we went, we never ran into a trash can that didn't have compost plastics and trash.
Okay, so what I've been talking about for the last 20 minutes has been implications of this TSD. Now let me take a step back and say, what happens? What happens in an embryo whenever we, either through a gene or through temperature, channel down a male pathway or a female pathway? Got some figures here for you. They're going to show you that every embryo starts its life with an indifferent gonad. A gonad that can go one of two ways. This indifferentiated gonad up here at the top, 4 weeks old. Here's another neat G Whiz Wild fact. This is an organ that can go one of two pathways that are so distinct from each other. It would be like saying, we've got an organ that can either become a heart or a liver. If you have the genes that guide femaleness, it becomes a testis. An ovary. It becomes an ovary. If you have the genes, I was already thinking one step ahead. If you got the genes that are for femaleness, you're going to be an ovary, okay? Now, how does that happen? Well, it's based on two ducts early on in the development. One of them's called the Wolffian duct, and the other one's called the Mullerian duct. The Wolffian and the Mullerian duct. These are two epithelial ducts that if we guide towards the testis, the Mullerian duct degenerates and the Wolffian duct is expanded. In contrast, if we go towards an ovary, the Wolffian duct degenerates and the Mullerian duct expands. Here's another way to think about it. This bipotential, sexually indifferent gonad can either take this yellow Mullerian duct and get rid of it. The hormone that does that is called anti-Mullerian hormone, AMH. Or, if we go the other way, we have the Wolffian duct. That the Wolffian duct is stimulated by testosterone. So without it, the Wolffian duct degenerates. And the Mullerian duct is retained. In the male, the Wolffian duct becomes the tube that's going to carry the sperm out to the ureter. It becomes the vas deferens, the epididymis, and the seminal vesicles. In the female, the Mullerian duct is going to become the oviduct, the uterus, the cervix, the upper portion of the vagina. These began as epithelial ducts in the gonad. Now, right beside, juxtaposed to the gonad. The gonads themselves are nothing but primordial germ cells that have migrated in from the yolk. Notice we have these sex cords that are in here. These sex cords are in a female. Let me go up this way now. In a female, they're going to be surrounded, the germ cells are going to be surrounded by these sex cords. This is eventually going to be a follicle. On the male side, what happens is these sex cords fuse together, and the germ cells go inside those sex cords. What I'm going to begin class with next time is I'm going to tell you about 4 particular genes that guide us down either going to an ovary or a testis. And then we're going to go into the function of male reproduction. Next class should be chapter 81, so if you want to look ahead, go ahead and do that. Does everybody have their tissues and paraffin blocks already? Anybody not? Has everybody got them in there? Awesome.
Not anymore. Yeah, Dr. James,
Yep.
do we need to store our slides some specific way?
Yeah, I'll get you a slide box. That way, yeah, you can just have them good and safe.
Can I get the
What's that?
Can I get the first